16.2 Digestion of food. The process of digestion is accomplished by mechanical and chemical processes. The buccal cavity performs two major functions: mastication of food and facilitation of swallowing. The teeth and the tongue, with the help of saliva, masticate and mix up the food thoroughly. Mucus in saliva helps in lubricating and adhering the masticated food particles into a bolus. The bolus is then conveyed into the pharynx and then into the esophagus by swallowing or deglutition. The bolus further passes down through the esophagus by successive waves of muscular contraction called peristalsis. The gastroesophageal sphincter controls the passage of food into the stomach. The saliva secreted into the oral cavity contain electrolytes Na+, K+, Cl-, SCO3-, and enzymes, salivary amylase and lysozyme. The chemical process of digestion is initiated in the oral cavity by the hydrolytic action of the carbohydrate splitting enzyme, the salivary amylase. About 30% of starch is hydrolyzed here by this enzyme. Optimum pH 6.8 into a disaccharide maltose. Lysozyme present in saliva act as antibacterial agent that prevent infection. The mucosa of stomach has gastric glands. Gastric glands have three major types of cells, namely: first, mucus neck cells which secrete mucus; second, peptic or chief cells which secrete the proenzyme pepsinogen; and third, parietal or oxyntic cells which secrete HCl and intrinsic factor essential for absorption of vitamin B12. The stomach stores the food for 4 to 5 hours. The food mixed thoroughly with the acidic gastric juice of the stomach by the churning movements of its muscular wall, and it called the chyme. The proenzyme pepsinogen, on exposure to hydrochloric acid, gets converted into the active enzyme pepsin, the proteolytic enzyme of the stomach. Pepsin converts protein into peptones and proteases. Peptide. The mucus and bicarbonate present in the gastric juice play an important role in lubrication and protection of the mucosal epithelium from excoriation by the highly concentrated hydrochloric acid. HCl provides the acidic pH 1.8, optimal for pepsin. Rennin is a proteolytic enzyme found in gastric juice of infants which help in the digestion of milk protein. Small amount of lipases are also secreted by gastric glands. Various types of movements are generated by the muscularis layer of the small intestine. These movements help in a thorough mixing up of the food with various secretions in the intestine and thereby facilitate digestion. The bile, pancreatic juice, and the intestinal juice are the secretions released into the small intestine. Pancreatic juice and bile are released through the hepatopancreatic duct. The pancreatic juice contains inactive enzymes trypsinogen, chymotrypsinogen, procarboxypeptidases, amylases, lipases, and nucleases. Trypsinogen is activated by an enzyme, enterokinase, secreted by the intestinal mucosa into active trypsin, which in turn activates the other enzyme in the pancreatic juice. The bile released into the duodenum contains bile pigment, bilirubin and biliverdin, bile salts, cholesterol, and phospholipids, but no enzyme. Bile helps in emulsification of fats, that is, breaking down of the fats into very small micelles. Bile also activates lipases. The intestinal mucosal epithelium has goblet cells which secrete mucus. The secretion of the brush-bordered cell of the mucosa along with the secretion of the goblet cells constitute the intestinal juice or the succus entericus. This juice contains a variety of enzymes like disaccharidases, example maltase, dipeptidases, lipases, nucleosidases, etc. The mucus, along with the bicarbonates from the pancreas, protects the intestinal mucosa from acid as well as provide an alkaline medium, pH 7.8, for enzymatic activities. Some mucosal glands, Brunel's glands, also help in this. Proteins, proteoses, and peptones, partially hydrolyzed proteins, in the chyme, reaching the intestine are acted upon by the proteolytic enzyme of pancreatic juice as given below. Proteins, peptones, proteoses, converted into dipeptides by trypsin, chymotrypsin, or carboxypeptidase. Carbohydrates in the chyme are hydrolyzed by pancreatic amylase into disaccharide. Polysaccharide starch is converted into disaccharide by the action of amylase. Fats are broken down by lipases with the help of bile into di and monoglycerides. Fat is converted into diglyceride with the help of lipases and is further converted into monoglyceride. Nucleases in the pancreatic juice acted on nucleic acid to form nucleotide and nucleoside. Nucleic acid is converted into nucleotide by the action of nucleases and further into nucleoside. The enzymes in the succus entericus acts on the end products of the above reactions to form the respective simple absorbable forms. These final steps in the digestion occur very close to the mucosal epithelial cells of the intestine. Dipeptides is converted into amino acid by the action of dipeptidase. Maltose is converted into 2 molecules of glucose by the action of maltase. Lactose is converted into glucose and galactose by the action of lactase. Sucrose is converted into glucose and fructose by the action of sucrase. Nucleotide is converted into nucleosides by the action of nucleotidases, and nucleoside is converted into sugar plus bases by the action of nucleosidases. Di and monoglycerides are converted into fatty acids and glycerols by the action of lipases. The breakdown of biomacromolecules mentioned above occur in the duodenum region of the small intestine. The simple substances thus formed are absorbed in the jejunum and ileum region of the small intestine. The undigested and unabsorbed substances are passed on to the large intestine. No significant digestive activity occurs in the large intestine. The functions of the large intestine are: first, absorption of some water, minerals, and certain drugs. Second, secretion of mucus, which help in adhering the waste, undigested particles together and lubricating it for an easy passage. The undigested, unabsorbed substances called feces enter into the cecum of the large intestine through ileocecal valve, which prevent the backflow of the fecal matter. It is temporarily stored in the rectum till defecation. The activities of the gastrointestinal tract are under neural and hormonal control for proper coordination of different parts. The sight, smell, or the presence of food in the oral cavity can stimulate the secretion of saliva. Gastric and intestinal secretion are also similarly stimulated by neural signals. The muscular activities of different parts of the alimentary canal can also be moderated by neural mechanism, both local and through CNS. Hormonal control of the secretion of digestive juices is carried out by local hormones produced by the gastric and intestinal mucosa. Calorific value of protein, carbohydrate, and fat. The energy requirement of animals and the energy content of food are expressed in the term of measure of heat energy, because heat is the ultimate form of all energies. This is often measured to as calorie or joule, which is the amount of heat energy required to raise the temperature of 1 gram of water by 1 degree Celsius. Since this value is tiny amount of energy, physiologists commonly use kilocalorie or kilojoule. 1 kilocalorie is the amount of energy required to raise the temperature of 1 kilogram of water by 1 degree Celsius. Nutritionists traditionally refer to kilocalorie as calorie or joule, always capitalized. The amount of heat liberated from complete combustion of 1 gram of food in a bomb calorimeter, a closed metal chamber filled with oxygen, is its gross calorific or gross energy value. The actual amount of energy combustion of 1 gram of food is the physiological value of food. Gross calorific value of carbohydrate, proteins and fats are 4.1 kilocalorie per gram, 5.65 kilocalorie per gram and 9.45 kilocalorie per gram respectively, whereas their physiological values are 4 kilocalorie per gram, 4 kilocalorie per gram and 9 kilocalorie per gram respectively.